Hey everyone, welcome back. It's Aditi Dubey here, and today we are zooming into the heart of where cancer gets its start, the cell cycle. Think of the cell cycle as the life story of a cell, how it grows, copies itself, and divides into two new cells. It's a beautiful, tightly controlled process that keeps our bodies running smoothly. But when cancer crashes the party, it's like someone rewrote the script. So let's break it down. First, the normal cell cycle. Picture it as a four-act play. Act 1 is G1, the growth phase. Cells get bigger and prep for the big event. Act 2 is S phase, where cells make a photocopy of its DNA, so each new cell gets a full set. Act 3 is G2, another growth spurt to double check everything is ready. And the grand finale, M phase, mitosis, where the cell splits into two. But here is the key. This isn't a free for all. The cell cycle has checkpoints, like security guards at each act, making sure everything is on track. Is the DNA damaged? Is it copied right? If something's off, the guards hit pause, fix the problem, or even tell the cell to bow out permanently. This is how our bodies maintain healthy growth, orderly, precise, no chaos allowed. Now, enter cancer. Cancer hijacks the system like a reckless driver smashing through roadblocks. Those checkpoints ignored. The normal rhythm thrown out the window. Cancer cells get stuck in go mode, dividing faster and faster without stopping to check if there is everything okay. What should be careful process becomes a frenzy unregulated division. They pile up, forming tumors because they have lost the memo that says, hey, only divide when the body needs you. It's not just speed, it's sloppiness. Each division can introduce more mistakes, piling chaos on top of chaos until you have got a full blown cancer situation. So, who is running the show or failing to? Let's meet some key players. Proteins like cyclins and P53. Cyclins are like the gas pedal. They rev up the cell cycle, telling it when to move from one phase to the next. They are supposed to rise and fall at right times, keeping the pace steady. But in cancer, they are often stuck on overdrive, pushing cells to divide non-stop. Then there is P53, the guardian of the genome. This protein is a checkpoint hero. It spots DNA damage and says, hold up, let's fix this. Or if it's too bad, time to shut down. In many cancers, P53 is mutated or missing, like a security guard who's fallen asleep on the job. Without it, damaged cells keep going, stacking up errors that fuel cancer's growth. These proteins are the tug of war between order and anarchy in our cells. There is a simple way to picture it. The cell cycle is like a traffic system. Normally, you have got green lights, red lights, and traffic cops, checkpoints, making sure cars move when they should and stop when they shouldn't. Cells flow through the cycle like cars on a highway, orderly and safe. But in cancer, it's a traffic system gone wrong. The lights are stuck on green. The cops are nowhere to be found. And cars, cells are speeding through, crashing into each other, piling up in gridlock, tumors. No rules, no stops, just chaos. That's what we are up against. This is where it all begins. Folks, the cell cycle is cancer's playground, and understanding it is step one to fighting back. Next time, we'll talk about those DNA mutations, the sparks that light this fire. For now, think about the traffic jam in your cells. How do we get the cops back on duty? That's the puzzle I wrestle with every day in the lab. See you soon.